as I said, my name is Simone Ferracina. I'm a research assistant for the Living Architecture Project at the School of Architecture, Planning and Landscape at Newcastle University. And I'm representing today the Living Architecture Project and its coordinator, Rachel Armstrong, who is also the stream leader in synthetic biology and biosystems for the Living Architecture Systems group. You're probably familiar, or very familiar, with the many collaborations that Rachel and Philip had for the Hydrozoic ground projects. Therefore, I'm going to introduce the synthetic biology stream and in a way interrogate the idea of architecture and metabolism by presenting a different project, which we're currently working on, and it's called also Living Architecture. As you probably know, a lot of Rachel's work revolves around the city of Venice. The Future Venice project looked at existing metabolic processes that could be harnessed and directed to produce calcareous reefs that would shore up the timber foundations of the city of Venice, or biofilms that could colonize plastic and build new complex soils. And when I say biofilms, you can imagine something like a microbial or bacterial city. And that is the start of the Living Architecture Project, an ongoing project funded by the EU Horizon 2020 Open Future Emerging Technologies, which is a collaboration between Newcastle University and the University of West England in Bristol, the Consejo Nacional de Investigaciones Científicas in Madrid, the University of Trento, and Explora Biotech in Italy, and Nuclear Systems Group in Austria. So what we're looking to achieve is a structural bioreactor wall made of modular living bricks. But what is a living brick or a bioreactor wall? They're programmable ecosystems that go beyond the representation of nature in architecture and rather directly incorporate living systems into materials, technologies and methods for their choreography of space. So if we go back to Venice, a little bit like being here and looking at the ceiling, and interrogate its stones, we find that these already are life-promoting infrastructures. That the natural forces in this anti-modern city already play a role in the production and negotiation of its architectural boundaries. We find bricks colonized by biofilm and seaweed, barnacles biocemented to stone, calcareous traces and accretions made by tube worms, Murano glass sintered to mixtures of rock and algae. Similarly, we strive to develop building infrastructures that work in sophisticated ways with living and lifelike systems, and do so in a contextual way. Designs and components that contribute positively to the complex system of ecological networks in which a building is situated. So the project will not produce one prototype of the living brick or the bioreactor wall and spread it across different ecological or socioeconomic contexts like the moderns would have done, but rather suggest a general set of technological recipes which can be contextually adapted and returned. But what are the basic aims and principles of this recipe? First of all, the capacity to harness and manage the biochemical capabilities of microbial consortia, performing dynamic physiological functions within a structured system that will be installed within an architectural context. So a house, a school, an office. So we aim to design a modular architectural bioreactor that is populated by a programmable microagriculture which can deliver specific biochemical products, usable biomass, fertilizer, polished wastewater, and functions generating oxygen and electrical power, extracting valuable resources from life. So obviously these are the very, very first prototype from a design standpoint, but they're working prototypes. Meaning that they already produce electricity. The system is based on the operational principles of microbial communities, both natural, the microbial fuel cell, and new to nature, synthetic consortia. The microbial fuel cell, or MFC, which is being developed by our partners in Bristol, is an electrochemical device whereby bacteria generate electricity through the oxidation of organic waste and renewable biomass. The chemical energy of organic feedstock is converted into electricity via the metabolic processes of microorganisms, such as which act as biocatalysts. So the microbial fuel cell consists of two compartments, the anode and the cathode, separated by a proton exchange membrane, or PEM. In the anode chamber, bacteria anaerobically oxidized organic substrate fuel, generating electrons and releasing protons. The electrons travel via an external circuit, the one on top, and the protons flow through the PEM to recombine in the cathode and react with oxygen to produce water. In addition, the principles of the photobioreactor, that is using light to cultivate phototrophic microorganisms, is combined with the MFC to produce a sequential hybrid bioreactor system. It is proven that algae can thrive in the cathodic environment of a microbial fuel cell, photosynthesize, and produce oxygen as a byproduct. The photosynthetically evolved oxygen is then utilized as at the cathode to enhance the oxygen reduction reaction for improved power generation and longevity. The living architecture bioreactor is therefore a complex managed microcosm or microecology, whereby defined sets of abiotic factors such as illuminance, temperature, pH, etc., are continuously regulated to create and maintain the desired environmental conditions that support the biotic system outcomes. Perhaps in a way not too different from Venice's natural negotiations of spatial boundaries. And finally, the final element to this recipe and defining ambition of the living architecture project is its incorporation of synthetic microbial consortia, MSC, into the built environment, expanding the scope of functionalities of the microbial fuel cell through design microbial communities that perform a la carte function as a kind of controllable metabolic app. Recent advances in omics allow the fast and detailed collection of the biological components, DNA, RNA, proteins, metabolites, required to understand the complex genotype-phenotype relationships in biological systems, including microbial communities. So the Living Architecture Project, in particular partners in Madrid, aim to better recognize the molecular basis driving the behavior of natural microbial consortia in order to develop orthogonal protocols for the design and engineering of synthetic ones. Therefore, through the systematic study of the living architecture microbial fuel cell and the available systems and synthetic biology tools, the project aimed to design a large array of synthetic microbial consortia with specific capabilities suitable to be included into building structures as microbial fuel cells. The project envisages two separate SMC modules, a cyanobacterial-based farm module exposed to the facade, and a bacterial heterotrophic-based labor module, interchangeable in place on the interior side of the building envelope. The farm module will supply easily metabolized carbon as energy source for the carbon module, while the labor module will perform the target biotechnological function, adding value to the whole system. Stability is achieved through metabolic cross-feeding. So where one organism synthesizes a compound that the other needs and cannot produce. So all the components of the SMC could be equipped with synthetic genetic elements, biobricks, designed for different functionalities, such as cleaning gray water or polluted air, or removing phosphate or nitrogen oxides. The labor module and the farm module are conceived as interchangeable devices which can be adapted to the specific needs of a building, for example, of producing biodetergents or biofertilizers. So this is the kind of brick we're currently working on, what we call a living brick. And again, it's not a particularly beautiful brick for the time being. But it does produce electricity, microwatts of electricity, but, volts, sorry. So it will be following a closed loop paradigm through sensors that will inform the central processor about the internal and environmental states of each reactor. And each reactor is, for instance, a brick. The range of external input parameters will include the chemical composition of growth media, the microbial consortium composition, and the abiotic drivers such as light, temperature, pH, and power consumption. Internal input parameters will be the operation and conditioned states of the neighboring reactors, which will be updated in parallel. So a wall built of many reactors, many of these bricks, will behave as a parallel processor with cellular automaton architecture, or a massively parallel reaction diffusion computer, a spatially extended chemical system which processes information by transforming an input concentration profile to an output concentration profile in a deterministic and controlled manner. The reaction outputs will include polished water, fertilizer, recoverable biomass, oxygen, and electrical power. At least that is the input. As mentioned earlier, living spaces thus become programmable ecosystem modules that can be customized to suit the available resources and needs of a community. And we'll be testing this technology in different contexts, working, for example, with the climate change challenges faced by the city of Venice and the energy and clean water needs of rural communities in Senegal, a scenario that we're working in collaboration with Massimo Lecquore of Thomas Ucciati in Italy. So the understanding of sustainability goes here beyond resource conservation and towards the incorporation of metabolic design and community into living spaces, moving the inert materials with a low toxicity profile to ecologically productive building components. Furthermore, the burning of fossil fuel is substituted by living technologies, by the wet burning of natural and synthetic metabolisms. That's it. Thank you. Sorry.